Today we're going to look at a couple of the really important gut bacteria that help maintain our mucosal immune system in the gut. The first one is Akkermansia muciniphilla. Our roundtable members are familiar with it. We've had it in our roundtable discussions for months, but we're going to take a deeper dive into it today and show you how some of our tiles that we're using for herbs and foods interact with it and what exactly it's doing in the gut immune system. So first of all, let's just click on this. What is it? So we know it's a mucin-degrading bacterium, and we've talked about mucin before. Its abundance in the intestinal tract is inversely correlated to several disease states. We know that it resides in the mucus layer, primarily of the large intestine, and there it's involved in maintaining our intestinal integrity. And it exerts a really beneficial action in metabolic diseases while fortifying our barrier function. It's associated with a decrease in inflammation, obesity, diabetes, and metabolic disorders. However, excessive colonization can break the balance between mucin secretion and degradation, and of course, that could damage the intestinal barrier. Now, you can increase your levels of Akkermansia by eating polyphenol-rich foods, and also by fasting because it feeds off your mucus, not your food. So we see here that it's primarily in the large intestine, but smaller quantities are also in the small intestine, especially in the lower parts of the small intestine. One question that comes to mind is if Akkermansia is consuming mucin, and we need mucin to maintain our mucosal layer, well, how does that benefit us? So here are some of the ways. First of all, it does maintain the mucus layer. Akkermansia uses mucus as its sole carbon and nitrogen source, producing mucin-degrading enzymes, and that does help to maintain the mucus layer. You know, you don't want too much of buildup, especially of older mucin and mucus. It's involved in nutrient cycling, so as it degrades the mucin, it's releasing other nutrients that are needed by other commensal bacteria. Remember, commensal bacteria means the good guys, the ones we want, the ones that help us. It structures the mucus layer. It strengthens the enterocyte integrity. It adheres to our intestinal epithelium and can strengthen that single layer, that single cellular barrier, those cells. It strengthens their integrity. And then, of course, it regulates mucus production. So, how exactly is it affecting our gut immune axis? We already said it helps to reduce inflammation, and it strengthens the gut barrier to prevent harmful substances from entering our bloodstream. But it also interacts with our immune cells to promote a balanced immune response. Here are some details on its functions and interactions. We already mentioned gut barrier enhancement, but it modulates some of our immune cells as well. Akkermansia reduces pro-inflammatory cytokines. Remember, cytokines are those immune messengers that tell the immune system to create inflammation or reduce inflammation. And it boosts regulatory T cells. Now, we haven't gotten into those yet. We'll talk about the T cells when we get into the adaptive immune cells. But basically, regulatory T cells are the cells that turn down the activity of T cells, which helps maintain immune tolerance and prevents excessive immune reactions. It's also involved in production of immunomodulatory molecules. So it produces proteins, lipids, short-chain fatty acids that directly interact with our immune system and help regulate immune responses. Now, these are context-dependent effects. So, while generally beneficial, sometimes It can increase inflammation in specific contexts. For example, when there's infection. And so we see that it has a complex role. Now here are some more comprehensive details. I'm not going to go through all of these with you. This is pretty technical, but I would recommend that you come in and take a look at the details of just what it's doing to enhance the gut barrier. And here's a table that summarizes the mechanisms along with the references. You can click on those to read the studies, the references. Here are details on how it modulates our immune cells, particularly reducing pro-inflammatory cytokines and promoting the regulatory T cells. And this is another table summarizing those. In terms of producing immunomodulatory molecules, these are immunomodulatory lipids, proteins, peptides, and short-chain fatty acids. Again, you can come in and click on those to read more about them if you want. And here we come up with the context-dependent effects. For example, when there's an infection like salmonella, the infection can be worse. And there can be strain-specific effects. And in fiber-deprived conditions, folks who are not getting enough fiber in their diets can actually wind up with Akkermansia exacerbating food allergies. And then we've got a bunch of citations. And here's another area. Again, I'm not going to go through this. It's a bit technical, but it's fascinating, I think. What role does it play in regulating host signaling pathways? Now, these are all hyperlinked, so I know most of you probably don't know what toll-like receptors are or nod-like receptors, but these are hyperlinks, so you could click on them and find out what they are. It's involved in calcium signaling. And here we see the IgA. We'll get into that later. Okay, so how does it affect metabolic disorders? Not going to go into that here because we are primarily focused on the gut immune system, but that will play a role in metabolic disorders, of course. How does it affect mitochondrial oxidation in the gut? That's important because, the mitochondria are what create the energy for our cells. And one part of the process for doing that is an oxidation process. Our Akkermansia is actually beneficial to this process in our mitochondria so that they can produce more energy. And as a result of that, the combination of what Akkermansia is doing in our mitochondria, we get improved lipid metabolism, the increased mitochondrial oxidations associated with enhanced lipid oxidation, and that helps reduce fat accumulation in the liver and ameliorates metabolic dysfunction associated fatty liver disease, something that's a bit too common these days. It reduces oxidative stress. Akkermansia treatment ameliorated oxidative stress-induced cell apoptosis, that's a type of cell death, in the gut. And by improving mitochondrial function, it can help reduce the production of reactive oxygen species. That's one of our tiles, gang. And protect cells from oxidative damage. It enhances energy metabolism. Improved mitochondrial oxidation leads to more efficient energy production in gut cells. And of course, that's going to support overall gut health and function. It's involved in gut-liver axis modulation. The increased mitochondrial oxidation in the gut-liver axis is associated with improved bile acid metabolism and increased L-aspartate levels in the liver. And those changes contribute to the overall metabolic improvements observed with Akkermansia treatment. and it reshapes the gut microbiota. The effects of Akkermansia muciniphilla on mitochondrial oxidation lead to the reshaping of the gut microbiota composition, and that suggests that the metabolic changes induced by it can have broader effects on the gut ecosystem. In a previous video, we talked about secretory IgA. So how does secretory IgA interact with Akkermansia? Well, first of all, through coding and retention. I'll just call it Sig-A for short. Sig-A binds to Akkermansia through interactions on its surface and coats it. This coating promotes its retention in the gut lumen and prevents it from penetrating the intestinal epithelium, maintaining a beneficial relationship with the host. Sig-A helps modulate the proliferation of Akkermansia by providing a controlled environment for its growth. This regulation ensures that it remains at levels that are beneficial to us without overgrowth. And by binding to Akkermansia, Sig-A facilitates immune tolerance towards this particular commensal bacterium. This interaction reduces unnecessary inflammatory responses while supporting Akkermansia's role in strengthening the gut barrier and promoting anti-inflammatory effects. Sig-A contributes to intestinal barrier integrity by preventing direct contact between Akkermansia and our epithelial cells, which could trigger immune activation. This helps maintain that healthy mucus layer where Akkermansia resides and performs its beneficial functions. And then it exerts cross-species reactivity, meaning it can bind to multiple commensal bacteria, including Akkermansia, and this broad interaction helps to maintain that balanced microbiota by supporting beneficial microbes and limiting the pathogenic ones. Again, it's important to note that while these effects are generally beneficial, there may be some context-dependent variations in certain conditions. So we see that it helps protect the gut barrier, but what about zonulin? If you'll recall, zonulin is a key trigger for opening up those tight junctions leading to gut barrier dysfunction. Well, it does interact with zonulin in ways that affect the intestinal barrier function and the gut microbiota composition. So we see here there are opposing effects. While zonulin increases intestinal permeability by regulating tight junctions, Akkermansia contributes to enterocyte monolayer. Remember, that's that single layer, the integrity of that, and it strengthens the gut epithelial barrier. Secondly, in regards to microbiota composition, increased zonulin levels are associated with reduced abundance of Akkermansia in the gut. Next, inflammatory status. Zonulin-dependent increased gut permeability leads to a pro-inflammatory status in the gut, whereas Akkermansia is associated with anti-inflammatory effects and improved metabolic health. As far as immune system modulation, zonulin-induced increased intestinal permeability affects the development of the immune system towards that pro-inflammatory state, while Akkermansia modulates the immune system, particularly CD8 T cells. Those are the killer T cells, potentially counteracting inflammation. And then as far as metabolic health goes, zonulin's effect on intestinal permeability can contribute to metabolic disorders, whereas with Akkermansia, it's associated with improved glucose tolerance. So we see here how having a healthy population of Akkermansia can offset some of the negative effects of zonulin. And if there's not enough Akkermansia Apparently there can be increased levels of zonulin. Now let's go on to explore dietary support of Akkermansia. We see that prebiotics help. The fructooligosaccharides, FOS, those are short-chain carbohydrates that are readily fermented by Akkermansia, and they stimulate its growth and activity. We find those in chicory, root, Jerusalem artichoke, certain fruits like bananas, and asparagus. Inulin is another prebiotic fiber that has positive effects on Akkermansia in studies. And inulin is found in wheat, onions, garlic, Jerusalem artichoke. And then, of course, the polyphenols. Certain polyphenols like apigenin, which we'll get to in a minute, and those found in green tea, red wine, berries, dark chocolate, can act as prebiotics indirectly by promoting the growth of other gut bacteria that stimulate Akkermansia. Then we've also got plant-based extracts like blackberry seed powder, pomegranate peel extract, urolithin A. Now that is a gut metabolite of the elagitannins, and it can potentially boost Akkermansia. Berberine promotes Akkermansia growth by stimulating mucin secretion in the colon. Now when it comes to ginger, ginger has a complex interaction. In some studies, ginger supplementation led to a decrease in the abundance of Akkermansia. Ginger treatment's been observed to reverse the decrease in the alpha diversity of gut bacteria caused by antibiotics exposure, which may influence Akkermansia population. So what it's saying here is be careful with ginger if you've been on a course of antibiotics. When you stop them, don't just start ginger. And then other dietary components include omega-3 fatty acids, capsaicin, that can indirectly support it because it promotes mucin production. Apple cider vinegar is associated with an increase in Akkermansia. That's probably because it contains acetic acid and polyphenols that positively influence the gut microbiota. And then blackstrap molasses, another one of our tiles. It seems to influence it through its prebiotic properties and nutrient content. Now what about fiber? Now at this point we're really digging into some of the tiles that we have in the round table, and so if you go through this you'll see more details on, for example, exactly what dietary fiber is doing. Lack of dietary fiber leads to increased abundance of Akkermansia in the gut, and that is associated with intestinal barrier dysfunction. Remember, you don't want too much Akkermansia, and that can exacerbate allergic reactions to food allergens. Something to think about with people who have a lot of food allergies. Are they getting enough fiber? Fiber-rich diet benefits. A fiber-rich diet supports a balanced gut microbiome, indirectly benefiting Akkermansia. Metabolite production. Akkermansia converts dietary fiber into short-chain fatty acids like acetate, propionate, and butyrate. All of those are hyperlinked, so you can click on them. These influence glucose and lipid homeostasis, affecting metabolic health. There are context-dependent effects. In fiber-sufficient conditions, Akkermansia helps reduce pathogen load. And we already mentioned in fiber deprivation, Akkermansia may contribute to increased susceptibility to mucosal pathogens. And then there are mucosal layer interactions. Dietary fiber helps maintain a healthy mucus layer, which, of course, Akkermansia uses as its primary nutrient source. And we already mentioned ginger. I'm not going to go through all the details here, but come in and see what ginger does, the potential synergy, the context-dependent effects. Let's move on to lemon. This is interesting. Lemon's far more powerful than we give it credit for. Now, we're going to talk specifically about lemon's polyphenols. That's what the LPP stands for. Those have a positive interaction with Akkermansia. Microbiome stability, lifelong intake of lemon polyphenols. And by the way, a lot of those are in the peel. Help maintain a stable gut microbiome structure in senescence-accelerated mice. Now, senescence simply means aging, similar to that of resistant mice. This stability may provide a favorable environment for beneficial bacteria like Akkermansia. Bacteroidetes firmicutes ratio. Lemon intake was associated with a higher Bacteroidetes and firmicutes ratio compared to control groups. Akkermansia belonged to the phylum Viricomicrobia, but its abundance is positively correlated with a higher Bacteroidetes firmicutes ratio. Moderation of age-related changes. Lemon intake moderated the age-related increase in lactobacillus species. This moderation of microbial changes may help maintain a balanced gut environment, potentially benefiting Akkermansia. And then the polyphenol effects. The dietary polyphenols, including those found in lemon, can stimulate the relative abundance of Akkermansia. And this stimulation contributes to the attenuation of several diseases. Again, obesity, type 2 diabetes, inflammatory bowel diseases. Now, the studies that show all this don't directly measure Akkermansia levels in response to lemon consumption. The overall effects of lemon polyphenols on gut microbiome health suggest the positive interaction. There are several classes of polyphenols in lemons that influence this beneficial gut bacterium. The flavonoids, the phenolic acids, the flavonoids, the flavonones. What about berberine? We kind of mentioned that already. Berberine has a significant positive effect on Akkermansia. Now you could come in here and really dig into it and see how it increased the abundance. It has an indirect promotion. Berberine increases intestinal expression of tight junction proteins and the thickness of the colonic mucus layer. There are metabolic benefits, and it's dose dependent. Okay, so what about licorice? In the round table, one of our tiles is deglycerized licorice. Does it have the same effects on Akkermansia as regular licorice? Let's take a look here. We do see that licorice has a positive interaction with Akkermansia, especially when combined with honey. Honey fried licorice is available online. Administration of honey fried licorice effectively improved gut microbiota imbalance with a more pronounced effect than regular licorice. It has a potential prebiotic effect, and it helps with the gut barrier. Both licorice and Akkermansia are synergistic in their anti-inflammatory effects. But what about the deglycerized? Glycyrrhizin in licorice is known to raise blood pressure, especially if you're using a lot of licorice. So that's why they remove it from licorice for people who want to use it for its many beneficial effects but are concerned about blood pressure. Regular licorice, which contains glycyrrhizin and flavonoids, has been shown to boost levels of Akkermansia in the gut. However, the DGL, which has had its glycyrrhizin removed, hasn't been studied for effects on Akkermansia. And given that glycyrrhizin might play a role in this increase, it likely doesn't have the same positive effect, making their impacts on this bacterium different. Now, if you want to look carefully at the research behind this, it's here. There are a number of studies and explanations I'm not going to go through all of that. You can always pause the video to read this if you want. We've got a comparative analysis between the two. So the bottom line on this is that if you specifically want to increase Akkermansia and you're not concerned about the potential of increasing your blood pressure, then you'd want to go with honey fried licorice. And I would suggest that you look up licorice in the database to see about the cautions in using regular licorice. Now on to milk thistle. We're very familiar with its miraculous effects on the liver, but what about in the gut? It has a polyphenol called silymarin that influences the gut microbiota by promoting growth of beneficial bacteria, including Akkermansia. Those polyphenols in milk thistle act as prebiotics. Silymarin also has a very strong anti-inflammatory property that can reduce gut inflammation. And since Akkermansia is associated with anti-inflammatory effects, milk thistle's properties may synergize with the bacteria's role in maintaining intestinal health. Also, milk thistle supplementation is linked to enhanced gut barrier integrity by reducing oxidative stress and inflammation. Again, there it complements Akkermansia and what it does. As far as metabolic health benefits, both milk thistle and Akkermansia are associated with improved metabolic health outcomes, such as better glucose tolerance and lipid metabolism. There are potential synergistic effects. Silymarin's antioxidant and hepatoprotective, that means liver protecting, properties may indirectly support Akkermansia by reducing systemic inflammation and improving gut-liver axis communication. And of course, that's crucial for maintaining a healthy microbiome. A lot of us are very familiar with using psyllium seed husk for gut health, but what specifically might it be doing with Akkermansia? First of all, it has prebiotic potential. Psyllium husk contains arabinoxylan. That's a gel-forming and poorly digestible fiber that acts as a prebiotic. Akkermansia doesn't directly metabolize dietary fibers like psyllium. However, it benefits indirectly from the short-chain fatty acids produced by other fiber-fermenting bacteria in the gut. So increased short-chain fatty acid production, such as acetate and propionate, supports Akkermansia, as these metabolites are beneficial for its growth and activity. So we're going to find that psyllium has an indirect positive effect. Psyllium supplementation alters the gut microbiota composition by increasing the abundance of beneficial bacteria like Faecalibacterium and Lactospira, which could create a favorable environment for Akkermansia. However, some studies suggest that psyllium might reduce the abundance of certain bacterial phyla, including Vericomicrobia, to which Akkermansia belongs, though this affects context dependent and more research is needed. It improves the gut barrier function. Psyllium increases stool water content and improves gut transit time, which may enhance the gut barrier's integrity. And since Akkermansia plays a critical role in maintaining the mucus layer and gut barrier, psyllium's effect on gut health could again indirectly support its function. And of course, constipation relief. It's widely used to treat constipation by increasing stool bulk and improving bowel movements. This improved colonic environment could indirectly benefit Akkermansia, since a healthier gut environment supports microbial diversity. As you may recall from previous class, vitamin A is the vitamin for the gut immune system. In terms of what it's doing with Akkermansia, it does influence its abundance and its activity. So we see that vitamin A supplementation can increase the relative abundance of Akkermansia. Vitamin A also enhances gut barrier integrity by upregulating certain proteins involved, such as occludin, which are crucial for tight junctions in the intestinal lining. Akkermansia, of course, also contributes to the gut barrier by promoting mucin production. So there seems to be a complementary relationship between vitamin A and Akkermansia. The immune modulating effects of vitamin A, such as increasing interleukin-22 expression, could further support Akkermansia's role in reducing inflammation and improving gut health. Vitamin A supplementation also increases overall gut microbiota diversity, which creates a favorable environment for Akkermansia to thrive. This modulation may involve pathways related to glycan biosynthesis and metabolism. Those are both important for Akkermansia's mucin-degrading activity. The interaction between vitamin A and Akkermansia may have therapeutic benefits for conditions like metabolic diseases, neurodegenerative disorders, and inflammatory bowel diseases due to their combined effects on gut health and systemic inflammation. Another one of our tiles is sugar. And sugar does interact with Akkermansia in complex ways that can affect glucose metabolism and overall gut health. First of all, glucose tolerance. It plays a role in improving glucose tolerance. Studies have shown that supplementation with Akkermansia can ameliorate abnormal glucose utilization, particularly at the transition from light to dark periods in mice. Insulin sensitivity. Akkermansia is associated with increased insulin sensitivity. The presence of this bacterium can help regulate glucose homeostasis and improve metabolic health. We've already heard about Akkermansia in helping with diabetes type 2. Metabolite production. Akkermansia produces short-chain fatty acids like acetate that can influence glucose metabolism. And these metabolites affect processes such as liver or hepatic glucose production and appetite regulation. GLP-1 secretion. Now, you guys have probably heard about this new weight loss drug, Ozempic. It's using GLP-1 processes. Here we see Akkermansia stimulates the secretion of GLP-1, which is glucagon-like peptide 1. And that's a hormone that enhances insulin secretion and suppresses appetite. So if you have enough Akkermansia, maybe you don't need to be taking Ozempic, which, by the way, has been shown to cause blindness. Not GLP-1, but Ozempic is causing blindness in a lot of folks. Signaling pathways. Akkermansia activates the P13K-AKT pathway in the liver, which plays a crucial role in glucose and lipid metabolism. That pathway is also the pathway that uses something called mTOR, or rapamycin. If you've heard of rapamycin as one of the new longevity supplements, it's because of its activity in that particular pathway. You can click on that hyperlink and find out more about that. That pathway also plays a role in cancer. As far as dietary effects, high sugar diets, of course, could negatively impact Akkermansia abundance, potentially leading to metabolic disturbances. Conversely, maintaining a healthy gut microbiome with adequate Akkermansia levels could help regulate sugar metabolism. Now that we've seen what supports Akkermansia and how it interacts with different tiles that we're using in the roundtable, it's time to turn towards what foods, medications, or herbs are harmful to it. Starting with medications. No surprise, antibiotics. Broad-spectrum antibiotics can reduce it by disrupting the gut microbiota. Some strains of Akkermansia muciniphilla do show resistance to specific antibiotics. For example, metronidazole. Antibiotic treatments often result in a decline in its abundance or alter its function. And then another concern is post-antibiotic supplementation of Akkermansia. That's been shown to aggravate colitis-associated colorectal cancer by damaging the gut barrier and exacerbating inflammation. So, the moral of that story is, once you're on those especially broad-spectrum antibiotics and you get off of them, that's not a good time to take an Akkermansia probiotic. Another problem could be anti-inflammatory drugs. Certain non-steroidal anti-inflammatory drugs, the NSAIDs, may indirectly harm it by altering the gut environment. As far as foods go, high-fat diets high in saturated fats, have been associated with a reduction in Akkermansia and may impair its beneficial effects. And low-fiber diets, we've already mentioned that. A diet lacking dietary fiber can reduce the availability of nutrients that support it and other beneficial gut bacteria. Fiber-rich foods like rye bran, wheat, alurone, on the other hand, have been shown to increase its abundance. And as far as herbs go, here we're looking at certain herbal medicines that are strongly antimicrobial. So, for example, oil of oregano. If you're taking a long course of oil oregano, strongly antimicrobial, so it makes sense that that could have a harmful influence on Akkermansia. Other factors, well, we already mentioned post-antibiotic reconstitution and excessive enrichment. Overgrowth of Akkermansia in specific contexts, for example, inflammatory bowel disease or certain cancers, may not be beneficial and could exacerbate disease progression. So, to summarize this, while Akkermansia is generally beneficial, factors such as broad-spectrum antibiotics, high-fat, low-fiber diets, and excessive enrichment in certain pathological conditions can harm its abundance or function. Maintaining a balanced diet rich in fiber and avoiding unnecessary antibiotic use are key strategies to support its health-promoting effects. Another one of our tiles is bloating. What role does Akkermansia play in this? It has a mixed but generally beneficial effect on bloating, depending on the underlying cause and gut environment. So the key points are a reduction of bloating through the gut barrier support by strengthening the intestinal barrier, by promoting mucin production and enhancing tight junction integrity. Well, that reduces intestinal permeability that can alleviate bloating caused by inflammation or irritation of the gut lining. In conditions like irritable bowel syndrome, where bloating is a common problem, Akkermansia has been associated with reduced intestinal permeability and improved gut health. And then, of course, the anti-inflammatory effects that can help alleviate bloating associated with inflammatory conditions. There's the interaction with the gut microbiota. By modulating the gut microbiome, it creates a balanced microbial environment that can reduce dysbiosis-related bloating. Its presence supports other beneficial bacteria. You may remember how when Akkermansia degrades mucin, it releases nutrients to feed other beneficial bacteria. Potential risks of overgrowth in certain contexts, as we've seen, excessive levels may thin the mucus layer due to its mucin-degrading activity, and that would exacerbate bloating or other gastrointestinal symptoms if the gut environment's already compromised. What clinical evidence is there? Some studies have shown that increased levels of Akkermansia correlate with reduced irritable bowel syndrome severity scores, including symptoms like bloating and abdominal pain. Pasteurized forms of Akkermansia have demonstrated efficacy in reducing colonic hypersensitivity an improved gut barrier function in animal models, which may indirectly reduce bloating. Do we find Akkermansia in animals? It's typically not considered a significant part of the canine or feline gut microbiomes. It is present in small quantities in some animals, but it's just not as abundant or prevalent in cats and dogs as in humans and rodents. So it's there. We don't know much about what it's doing there. It may be beneficial for managing obesity in pets or impacting inflammatory bowel diseases. I'm not going to go through this, but you can come in the database and find out what its role plays in colon cancer. Now we're going to finish up with Akkermansia here, and we're going to move on to bifidobacteria in the next video.